Thank you, Richie. Good afternoon, everyone, and welcome to the March GT Bob Green Industry webinar. Thank you for joining us today. Our first speaker is Dr. Ruchika Kashyap, who is an assistant professor in the Department of Plant Pathology at the University of Georgia. She investigates plant disease challenges with a focus on fungal and Oomycetes pathogens in indoor and urban production systems. Her research leverages tailored strategies driven by needs assessments and strong stakeholder collaborations. Dr. Kashyap's goal is to develop sustainable plant disease management solutions that directly benefit producers while bridging the gap between farm and fork through impactful science communication. Additionally, she leads an extension program that fosters a two-way learning process, translating research into actionable solutions for the agricultural community. Welcome, Ruchika. The floor is yours.
Thank you, Dr. Benisi. Thanks for the introduction. I hope you all are able to see my shared screen.
Yes.
Looks perfect.
Perfect. Hello, everyone. Thank you for joining. I'm Ruchika Kashyap, and I'm a newly hired assistant professor and extension specialist of urban and controlled environment plant pathology at University of Georgia in the Athens campus. And for today's webinar, we are going to explore plant disease challenges and their management in urban and controlled environment settings. So for my talk today, we are going to understand some basic concepts of plant diseases and their management, and then focus on urban and controlled environments, where I will show you some new systems that I have come across in Georgia while surveying for plant diseases and the common challenges that might be out there. And what are the common disease challenges and how we can best manage them, and how we can incorporate integrated disease management strategies in indoor productions too, just as in, we apply it for field production systems. So starting with the basic slang or thing that says plants get sick too. But what is a plant disease? Plant disease is anything that is not normal to a plant. So it leads to, or it's caused by impairment of the normal functioning and results from continuous irritation by either a pathogenic or a non-pathogenic agent, which might be an environment factor or a non-infectious factor. It usually progresses over time, and that is the cause of symptoms that we see on plants. And the symptoms can really vary from, below ground symptoms or the symptoms that we see on roots and crowns, which includes root rots, crown rots, nematode issues. We're seeing some galls or deformed roots, and this can be caused by either, again, both pathogenic and non-pathogenic issues or problems like overwatering or poor planting, or fungal and other biotic issues as well. So the second type of symptoms are mainly caused above ground, either on the foliage, stem, or fruiting parts, and these can be leaf spots, cankers, fruit rots, etc. There are third kind of symptoms which are classified as systemic, that is, it is internally present inside the body of the plant. And these are usually caused by systemic pathogens such as viruses and phytoplasmas, and they infect the xylem or phloem tissues, etc. But how do we figure out what's wrong with a plant? In order to figure out what's wrong with a plant, you really need to be detectives. You need to ask any possible question that can lead to a plant disease. Questions such as, is this normal for my plant to look like this in this particular season? Sometimes the symptoms can be just natural senescence. So you really need to figure out what, when, and how are you seeing these symptoms. What is the plant care history? You need to know what did you apply, when did you apply, and is there a correlation between any of the cultural practices that you have used and the symptoms that you might be seeing now in order to figure out and decipher really deep on what's going on with your plant. Is there a pattern to your symptoms that you're seeing? Usually, abiotic factors, which might be nutrient deficiencies or waterlogging or any such environmental issues, they lead to uniform symptoms throughout. But biotic symptoms are usually random. As an example, if we were sitting in a room and I had some flu or cold symptoms, so the people most likely to get flu or cold symptoms will be the front row folks, which are closer to me, or folks who are the immune system, who have a little weaker immune systems than others, right? Not everybody will get flu in the whole room. So that is the case with biotic diseases. When it moves from plant to plant, not all the plants will be usually affected by plant disease. That's how these are some of the things you can differentiate between biotic and abiotic factors. You also need to pay attention on what kind of symptoms you're seeing. Depending on what kind of symptoms you're seeing, you can really decipher and differentiate on whether it's a fungal cause or an abiotic cause or a bacterial pathogen or a viral pathogen, depending on certain characteristic features that you might be seeing. The other thing that is really important to notice is the sign or the visual presence of pathogen or mold on the plant, because that is a characteristic feature that it is a biotic cause that might be causing this disease. So, in summary, you really need to think about any possible change or any possible links in your cultural practices that can help us narrow down to the cause of the symptom that you're seeing on your plant. We also need to understand the disease triangle, that a disease only happens when a susceptible host comes in contact with a virulent pathogen, and then a favorable environment is already present, and then the disease will occur. This is a very infamous disease triangle in basic plant pathology, but it comes back again and again because the basic concepts of how disease appears lies in this disease triangle. So, what is a susceptible host? It can be a stressed or an injured plant, or it can be a susceptible plant. We have resistant varieties out there for some diseases, but not for all diseases, right? So, if a susceptible plant is planted or it is stressed due to other abiotic factors, it's too much of waterlogging in there or too much of drought stress that's making the plant weak, so then it's more susceptible to getting pathogen infection. And the second thing is, conditions that we were talking about. Wet foliage, what's the soil condition like? pH, what's the humidity like? How closely planted are the plants? And what's the air circulation like? And all these factors, depending on what pathogen is present and is capable of causing the disease, can lead to a disease. But if two of these three factors are present and we can eliminate the third factor, meaning we make the environment unfavorable, or we plant a resistant variety, but the environment is favorable and the pathogen is there, we will not get disease. So that is why again and again the disease triangle is highlighted, so that we who are managing different plants can make sure we either plant early, plant late, depending on the weather conditions, depending on where you are handling your plants or what area you are in. You can manipulate these conditions and prevent the disease from happening in a plant. Now, coming back to causal agents of disease, as mentioned, they can be biotic. And in biotic diseases, usually four common pathogens are the causal agents of biotic diseases, which can be fungi, bacteria, viruses, and nematodes, apart from other diseases like there are plant pathogenic plants as well. And abiotic diseases include air pollution, herbicide drift, nutrient imbalances, water stress, too much of water, temperature. All these things can also cause diseases in plants or symptoms in plants. So, to diagnose and manage a disease, it is really important for us to first determine whether the disease is biotic or abiotic. Only then we can, manage it and control it. And injury to plants is typically a one time incident, such as shredding of leaves by hail, and, maybe wind currents happen or a branch break, and there was a deer and then it broke the branch or something. But plant disease is different from an injury. It is actually caused by living organisms such as fungi, bacteria, viruses, and nematodes. And it usually results in continuous progression of the disease symptoms over time. So there are things that you can use to visually differentiate between abiotic and biotic symptoms as well. Talking a little bit about the causal agents, we'll talk about fungi first because they are by far the most important group of plant pathogens. And there are over 8,000 species that can cause plant disease, but not all fungi can cause plant diseases. And majority of fungi are actually not plant pathogens. So these fungi primarily survive on non-living organic matters and are called saprophytes, but there are some fungus which need living host and are called obligates. So there are different diseases that symptoms that fungi can cause, starting from root symptoms, leaf symptoms, and you can also see signs of pathogen in some cases in some fungi like rust and powdery mildews. You can literally see the spores of the fungus on the leaves. And then it can cause internal discoloration of stem as well, like in fusariums. It causes internal discolorations when you split open the cross sections, and it can lead to deformed structures as well. So there are a range of symptoms that fungus can cause. But in order to differentiate what fungus has caused what symptoms or what's the causal agent behind these symptoms, plant pathogens used characteristic symptoms that typical fungi produces. It can be at microscopic level. For example, usually fungus produces thread-like structures where a single thread is called a hypha. And the hypha in combination or many hyphae in a mass are called mycelium. Most fungi produce hypha, as one of the pictures shown here. And this is what we also look under the microscope to see what is the structure or integration of Hyphal-like. For example, there is a pathogen called Rhizoctonia, where the characteristic feature of Rhizoctonia is the configuration of cross walls of hyphae. They are at 90 degrees. Whenever the hyphae are at 90 degrees, it is an indication or a characteristic feature of Rhizoctonia, and where diagnosticians use this to identify the pathogen Rhizoctonia, which is a common cause of root rot in different plants. Another example is fungi usually produce fruiting bodies. As you can see in here, at the lower surface of the leaves, they are these small dot-like structures, black blobs in this photo. These are called fruiting bodies. Now, within this fruiting body, a fungus produces a lot of spores. Some fruiting bodies are asexual in nature, some are sexual in nature, depending on how in the reproduction cycle they came in. But what is important to note is this is a sign of the pathogen presence, and it is an indication that this leaf, whatever we are seeing in here, is most likely a fungal pathogen that has caused this, and it's not abiotic. Now, we were talking about fruiting bodies a lot. There are these are another characteristic features under the microscope when we look at some of the fruiting bodies. And this fruiting body is a picture of a Cleistothecia of a powdery mildew fungus called Uncinula. So you can see, depending on the shape of the fruiting body, Cleistothecia is usually a closed-shaped fruiting body. You can identify and classify what fungal species or what fungal genus might be affecting the crop. Another example is, spores. Fungus produces a lot of spores, and the spores can have different shape. They can be segmented or not segmented, or some might have cut. Some of them are colored and not colored. Some are multicellular. So this is the spore of the fungus Fusarium, which is a common wilt-causing pathogen, and they are usually colorless. This picture, even though it appears blue, it might be due to staining. So its banana shapes of these cell are characteristic features, and they are multicellular. So that's how we differentiate the pathogens under microscope. Another example is a picture of fungus Thielaviopsis. Again, it produces multicellular spore, but they are dark pigmented, as you can see in these pictures, which is another example of how we can differentiate the spores. This is the last example I have in the fungal spore pictures. This is not a fungal spore, but a Oomycetes spore, which is another common cause of plant diseases. And it is a fungal-like organism. It was previously classified under fungus, but now it's no longer a fungus anymore. But pathogens like Pythium, Phytophthora, which are common soil-borne pathogens, even downy mildews, are classified under Oomycetes, also called as water-loving pathogens. So this is a typical spore, oospore. This is a resting spore that the fungus produces. And spores of this type can survive up to 40 years in soil. So this type of structure is produced by those Oomycete pathogens, and we also can differentiate these. Now, coming to the second cause of plant diseases is bacterial diseases. Now, bacterial diseases, unlike the fungal pathogens, do not produce, or in most instances, do not produce any spores except some resistant structures that they produce. Now, the infections caused by bacterial diseases usually are tissue disintegration, water soaking, some ooze that you will see from the plant coming out. Excuse me. And then usually they occur when the plant wetness is high, when there is dew on the plant, there is high humidity, and warmer temperatures favor these disease. And bacterial cells usually spread by water splashing, tools, hands, and insects. And here is the plate of a bacteria where you can see the streak of bacteria and single colonies of bacteria as well. Now, this is an microscopic picture of bacterial cells, and bacterial cells are extremely small. Even when magnified to like 400 times their normal size, they are difficult to distinguish as individual objects. Often they need staining to see them. They are usually rod or spherical shaped, and most plant pathogenic bacteria are rod shaped. Another group of pathogens that we talked or discussed about is viruses. Viruses are another common cause and very important cause of plant diseases. While bacteria are small, but viruses are even smaller. Bacteria can be seen with a light microscope, but viruses can typically only be seen with an electron microscope. But viruses are honestly much simpler structurally because they just have some DNA or a genetic material which is surrounded by a protein coat. So that's all they have. They don't have their own machinery, so they use the host machinery, invade the host machinery to cause diseases, just like viruses cause diseases in humans. It invades our immune system and stuff like that. And usually the kind of symptoms that we see in viruses are leaf mottling, curling, stunting, twisting of leaves and deformations and stuff like that, as you can see in these pictures. And viruses are usually, they need vectors to be transmitted. And most of the viruses are insect transmitted viruses like whiteflies, aphids. They transmit the virus, but viruses can also be mechanically transmitted through tools and, while we are working through grower handling as well. As I mentioned that it can only be seen through electron microscope. This is a picture of a viral particle magnified to about 100K times, 100x. So, sorry, 100,000 times. So, plant virus can be really rigid as well. So last cause is the nematodes. Nematodes are usually soil-borne pathogens, but they are, and they can cause symptoms like knots in the roots, root knot nematode, which is usually the cause of these root knots, and they can also lead to excessive plant damage. Now, most nematodes are beneficial, but some attack plants. They are mostly in the soil, but like I said, some can also be found above ground. They live on roots using stylet. Now, stylet is their mouth part, which basically they use for puncturing in the roots. And, that's how they feed on the roots. And they cause loss of roots, stunting of plants, and their management is basically you really need to keep your soil healthy, proper plant selection and rotation as well. Now, coming to the abiotic problems, because we talked about the biotic problems, the main causes, which were fungal, bacterial, viruses, and nematodes, and in addition, oomycete problems, such as caused by Pythium, Phytophthora, and downy mildews. Most problems are abiotic. So, abiotic problems are basically non-pathogenic disorders, which are either caused by environment conditions or cultural practices, which can be, as mentioned, too much water, usually planting in a wrong site, planting a wrong plant, or inappropriate placement, like where in your garden or your landscape have you placed that plant? Is it at a level where the soil is, it's at a lower level of inclination, or where is the placement? Is it towards shady or is it in the sun? So where the placement is also important. One of the main nutrient deficiencies, one of the main abiotic cause of discoloration of foliage is nutrient deficiencies. Common discolored symptoms include yellowing or chlorosis and reddening. So noting the pattern of yellowing can give you some clues about the normalities. Now, there are a lot of resources available on the web to visually diagnose the nutrient deficiencies. And I came across one resource, which I took from this article here, where they basically show you the kind of nutrient deficiencies, both macro and micronutrient deficiency, on which leaves they appear, newer leaves and older leaves, and what kind of symptoms you can expect from these deficiencies. On the right picture here, this is mainly occurring along the margins and is due to lack of molybdenum, which is a minor plant nutrient. So this is just to show you that the symptoms can be really similar to what you see in biotic conditions. But if you kind of figure out where in my plant, is it younger leaves, older leaves, when I'm seeing the symptom, what kind of symptoms, you can diagnose the symptom that we're seeing. Some symptoms like herbicide exposure can lead to curling and cupping of the plants. So you have to be really careful, with the increase in number of herbicides being used on lawns by homeowners and lawn care professionals. The non-target plants, these herbicides have increased and their exposure to non-targets have also increased. And this is the typical symptom of herbicide exposure too. And there can be other symptoms as well. This is another symptom of pollutant or pollution damage. Reddening symptom of this prunus are due to severe ozone injury. So just to show you another example of how, symptoms can be abiotic and not biotic. Another example of cold injury, as you can see on spruce, the new branch tips began to expand. The tree experienced a period of cold that was severe enough to kill the growing tips. Now, to correctly identify this kind of injury, you really need to know during what time of the season you first saw these symptoms. So that's why it is so important to monitor the history of your crop production or your plant production throughout so that you can correlate with what kind of symptoms you're seeing. This is the example of a heat injury, which is caused by temperature on rhododendron. Due to high temperatures, there is tissue damage you can see here as well. So now, summarizing the abiotic factors, there's so many abiotic factors: cold injury, heat injury, herbicide damage. There can be some mechanical damage as well that can be caused because of mowing or some other practices that we do on our farms, backyards, or landscapes, or wherever we are growing our crops. So you really need to see the patterns of abiotic damage as well. So they are usually uniform, cover more than one host because it does not matter what host it is, and they're usually widespread as well. So these are some of the things that you can help you narrow down abiotic patterns. The other thing to consider is the disease cycle of the pathogen. So the disease cycle of the pathogen is basically its survival, how it survives, how it disseminates, and then what kind of symptoms you are seeing, right? So if there is a pathogen surviving in your field already, so when favorable conditions will happen, it will germinate and cause first symptoms and then try to spread across. So now when it disseminates, it can disseminate through your tools, wind, water splash, rain, depending on what kind of pathogen is there. So you really need to also first know what is affecting your crop, either it is abiotic or biotic, and then know, how it can spread. And really, prevention is the key for management. That's what we are going to talk about here, that managing diseases, if you prevent the disease through cultural practices, it is the best we can do in terms of management, and then think about biological or chemical strategies for later purposes. I'm sorry. So in cultural practices, exclusion, make sure you have disease-free seeds, you use clean garden equipments, especially if they are shared. Clean the garden equipments after every use and before every use. Also, make sure that when you start any cultural practices in your garden, backyard, landscape, or in your farm, start where stuff is really healthy, because then there will be less chance of moving from disease to healthy because you've already done with your healthy areas and then you're working on your diseased area. So these are some best management practices that you really need to take care of. And then eradicate. Sanitation is the key for any managing any plant disease, basically. Remove diseased plant parts, rotate when feasible, mulch plants and stuff like that. Use resistant varieties if available. And then come to protection. When some diseases are inevitable, there is no denying the fact, right? They're favored by weather patterns brought from air currents. So use preventative strategies, spacing, staking, improved soil, take care of your soil health. And then chemical fungicides should be basically the last option. And use this integrated disease management approach to avoid resistance and better care of your plants as well. Now, talking about urban and controlled environment ag. So far, I talked about really basics of plant pathology, what are diseases like, what are the common causal agents, what are the different management strategies, and how diseases can spread, and why it's important to diagnose the disease, right? But now I'm going to talk about urban and controlled environment ag and how it's unique and how it's different from field practices and what I've seen in Georgia so far. So we know that the climate is changing and urbanization is happening. So we need diverse, resilient year-round food production, and controlled environment or indoor ag or urban ag is a way to do it. So most of the crops that are grown in these indoor ag controlled environment settings are vegetable crops, which are tomatoes, lettuce, cucumbers, peppers, and some fruits like strawberries as well. But this production nowadays have moved from urban, suburban to rural areas as well, especially with the concept of people's garden, community gardens, where people are coming together and joining hands to provide healthy food products as well. When we categorize the CEA, they really expand from greenhouses to vertical systems, and they can be low and mid tech like hoop houses, high tunnels, and to high tech like hydroponics, aeroponics, aquaponics, and things like that as well. So far, there are a lot of companies that have invested in, all these hydroponic, aquaponic type controlled environment production in Georgia, and every day a new company comes in because this is, like I said, the future and a way to produce healthy food. But this also comes with challenging diseases, and diseases are similar diseases that we see out there, but the management might be slightly different, and that's what we'll talk about here. So, just highlighting UGA's efforts for controlled environment ag. So, UGA has built a controlled environment ag team where we have 5 researchers working in this controlled environment area, starting with Dr. Renito Ferrazzi, who will work on the CEA crop physiology, me as a CEA plant pathology, Dr. Song as CEA phenomics, Dr. Andrew Ogden, the CEA breeder, and Dr. Eric Schuler, the CEA entomologist. And we are also focusing on building new infrastructure so that research can happen, need-based research can happen at both Athens and Griffin campuses of UGA. So talking about CEA system and crop diversity, really, they are really diverse. It can vary from a rooftop farm, a vertical farm, indoor garden to community garden. And then, like I said, hoop houses to different structures as well. If we want to classify controlled environment ag, they can be classified based on 2 methods, either cultivation methods, which is basically soil cultivation, or you're growing in a completely water or substrate based cultivation, or you are misting the water and then, or you have fishes along with water that are circulating. In the cultivation format, on how the format is your system inside a greenhouse, or is your system vertically completely indoors or stuff like that? So there are different ways we can classify it. Now, plants get sick in CA too. You can see some of the pictures here that I clicked in some facilities, and this is from an eGrow article by Dr. Woodward. You can see the difference between healthy and diseased roots in a deep water culture system and other diseases, and we'll talk about some diseases that I'm seeing in these CA systems. But before moving on, just correlating back to the plant disease triangle that we talked about earlier, now how does it differ in a CEA? Now, host, susceptible, and stressed will remain the same. The pathogen should be aggressive. Now, the environment here might vary because it's under an indoor system and the systems of production is different. But again, depending on the environment, like what's the light intensity, what kind of light you're using, what's the circulation like, is it more humid inside? The problems and diseases that we commonly see might vary, but the concepts remain the same. That's why I brought this disease triangle back again. And again, we need to be a detective to see how do we manage this disease. So my lab, Urban and Controlled Environment Pathology Lab, is specifically focusing on urban and controlled environment system. And I really want to generate site-specific crop-based information because there is a lot of information that is lacking in this sector, and stakeholders need updated information on what diseases are present, and so that I can work on those need-based solutions and help them out and then disseminate my efforts. So, what system diversity I've seen so far in Georgia? So I've seen different hydroponic systems. On the left, you can see a deep water culture, which is basically a big tub with water and nutrients are supplied in that tub. And then plants are grown, usually leafy greens are grown in these foam trays that are just, swimming on the water. And then the second type of system is NFT, which is nutrient film technique, where water flows through these nutrient films and plants are grown in these holes that are out there. The second type of hydroponics I have seen is substrate-based hydroponics, which is basically not soil, but coconut coir-based substrate that is used for growing, and then the water is intermittently supplied through drips. And these are Dutch bucket type or back to bucket type systems, and this is tomato production that's happening. And this on the left, there was a grower who really wanted to start hydroponics, and he was having some disease issues, as you can see, wilting. But it was we felt like it was abiotic because we did not recover anything in our lab. But what I want to point out is even in the center, this is a big company that's already in Georgia and is doing acres and acres of hydroponic tomato production. So from small growers to big growers, they are, adopting hydroponics, and they are even trying cooker beds, apart from tomatoes in the substrate or based or grow bag type production system. Lettuce is really a common thing in hydroponics. And then this is a hydroponic facility I visit where they combined substrate and hydroponic NFTs. So in the first layer, it is substrate based, but the subsequent layers are, just water flows down through gravity. So people are getting innovative, and there are different ways to grow hydroponics and in hydroponics. And then there are these microgreens and seedling productions that has also been coming up as a part of these hydroponic and controlled environment facilities. Another system is vertical towers. As you can see here, these are vertical towers where through pumps, water is supplied intermittently, and some of them are aeroponics where water is misted as well. But this is another vertical type system, but it is basically not a plus PVC type system here. So just to highlight how different growers use different techniques of growing. High tunnels, coming back to the original structure of growing, and then, or the basic low tech, mid tech structure of growing organic high tunnels. And then community gardens. So we've been surveying across community gardens too. So what symptoms I have seen? I've seen root symptoms, foliar symptoms, and all kinds of symptoms. As you can see here, root tip discoloration, complete root browning, some foliar tissues, and hair crown browning, and then the seedling emergence issues as well. So talking about biotic and abiotic issues, just like other facilities, hydroponics also have abiotic and biotic issues. One example of abiotic issue is a tip burn, which is usually referred to as there are two types of symptoms you can see: inner kind of structural symptoms and then outer symptoms. Usually this is referred to as calcium deficiency, but there are multiple causes, abiotic causes like combination of airflow, low humidity, which exacerbate these conditions as well. Coming back to pathogen challenges in CEA systems, Botrytis is a is a common pathogen, which is basically gray, fuzzy appearing spores, also known as gray mold, that appears on tender, freshly injured tissues. And this is really caused by high humidity and condensed plant material. So keep plant canopy dry, make sure there is sufficient ventilation and low humidity and sanitation. Second is powdery mildew. Powdery mildew is superficial growth of the fungus, and usually it starts with irregular chlorotic spots. They first appear on the upper leaves, but then this is also caused by high RH, and adequate plant spacing, proper ventilation is basically, or applying water directly to these leaves because it does not like free water could be a management practice. Another important disease that I'm seeing is the blackleg disease, which you can see here. This is from, on the left, this is from a publication from Florida, where they show that Plectosporella is cause of this blackleg in basil. I'm seeing similar symptoms in one of the basils that I sampled from one of the growers I visited. You can see blackleg symptoms. So it's something to study more on or research more on. Now, lastly, the root diseases are so common as well. So root diseases are mainly caused by Oomycete pathogens such as Pythiums in hydroponics, and they are basically water-loving pathogens. So they have these spores where they have two flagella and they can swim, and hydroponics is all about water. But depending on the temperature conditions, pythiums like to certain different temperature conditions. So there are recommendations of, cooling down the water temperature to manage pythiums, but you really need to know what pythium species is present in your system. So make sure that you go and diagnose what disease species you are dealing with. So the sources of contamination, honestly, they can come from anywhere, from outdoor streams, with shoes, with tools. They are soil inhabiting, they love water. And managing, I would say this is a long slide, but honestly, it's sanitation. So make sure you don't reuse your rooting mixes, you sweep your floors, if you treat your surfaces with sanitizers, make sure that all surfaces are not treated the same. This is a very nice e-grow article on root diseases in hydroponics that can be really helpful as well. I know that I think we are almost at the end of the webinar, so I'm gonna skip really quick and come to the last disease that I usually see in hydroponic systems is Thielaviopsis, similar to Pythium. It causes root browning, and then it spreads in water and soil via spores, and fungal gnats can also spread the spores. And again, for controlled environment systems, the best management practices is basically sanitation. So, summarizing the important management practices in CEA, use resistant tolerant varieties whenever possible. Sanitation, disinfect your tools, services, identify what pathogen you are dealing with, stop the spread and dispose the areas, quarantine areas, separate it out from other areas. Use fungicides or biofungicides for indoor production systems. Not a lot of chemicals are labeled, and biofungicides could be an option for you, but fungicide is the last option. And make sure you read the labels because for indoor production system, they are usually not applicable. So control your insect vectors and optimize your environment and crop and, against pathogen as well. So adopt this systematic disease management approach in order to manage diseases of hydroponics and controlled environment and urban systems. Basically, avoid any entry points. Take care of any entry points like seeds, media, water, benches, surfaces, people that could lead to introduction of pathogens. Make sure you manage the dispersal points, that is air, water, people, and mechanical dispersal. And then make sure You control the factors that could aggravate the dispersal, like humidity, nutrients, airflow, temperature, densities. And if we adopt this systemic disease management approach, it is a very good method for managing plant diseases in urban and controlled environment conditions. So I'm going to wrap up. So divide your strategies into preventative, diagnosis, and then management. This is again the same. Make sure you sample and send samples to us. I do a survey. I'm doing a survey across Georgia. So if you know about any growers that want their samples like water, soil, substrate, plants tested for any plant pathogens, please reach out to me and I can help you diagnose different pathogens that might be present. And I'm just skipping these slides and moving on to, thank you.
Thank you, Ruchika. This was very informative and rich with information that we can all digest when we have time, which is quite a bit. It is important to recognize that this is a very complex environment we're dealing with. And so I'm opening the floor for questions. And you've covered a lot. You really did. It's amazing how complex the management of diseases could be, just from diagnosis to recognizing different hosts to different symptoms in different hosts to, yeah, lots and lots of different things.
Yeah, that's what my idea was, Dr. Panisi, as well through the webinar. I wanted to first focus on, how even the common diseases can be challenging to identify, between abiotic and biotic. And what you really need to consider is just take care of your plant history, right? Just make notes of everything. That's the most important thing. Once you know, you'll be able to correlate. Oh, that's what I did, and this is what happened with the weather. So it's so important to really correlate things, and prevention is the key. So if we have that history noted, so definitely we can prevent a lot of diseases. And then I wanted to correlate with controlled environments. As you can see, I was rushing through my presentation at the end because I know it's a long webinar, but I wanted to correlate and show you how complex these controlled environment systems can be. We say hydroponics, but even in hydroponics, there's so much variability as well. So where the pathogen is growing, how it's spreading, is it spreading vertically? Is the vertical spread possible? Or if it's a horizontal system, so it really, there's a lot of challenges here in terms of management. So hope you were able to get some insights on management, and feel free to reach out to me with any questions even later as well.
Oh, absolutely. Every time I listen to a pathologist talk about diseases, and it's like, oh, I forgot this. Oh, there's something else to learn. So absolutely, yes, we never stop learning. And when you were talking about the factors that can spread diseases and problems that can occur, I was thinking about a problem that has been happening with boxwoods in the landscape. And several years ago, it was particularly impactful in Georgia. And we went to one client in Buckhead in Atlanta, and they have these really old, very healthy for many years boxwoods. And it turns out that it was the favorite pet, the cat, that was spreading all the spores as she was going up and down that favorite little path of hers. And you could just see, and her tail was just right there, perfect brush, just spreading the spores like intentional. So yes.
Yeah, so that's what it ends up to be, right? Thinking about what might have happened. Because sometimes the main question the growers have, you know what, I'm doing everything the same. Still, this came up, or I'm seeing this. There is definitely something that changed, either in the disease triangle, either in the environment or the host or the pathogen, or the dispersal factors, that we were talking about. Something changed that aggravated the disease.
Absolutely. If there are no more questions or no questions from the audience, I just remind you, if you come up with some, I think Ruchika would hang out with us till the very end, and you can always ask her. With this, thank you again, and we are going for a break.
Thank you so much.